안녕하세요. Hello, everyone. I hope you all can see me and hear me clearly. So my name is Jane, okay, and I'm the field application scientist from GenScript. So thank you so much for joining me here today. Okay, so for today, right, I would like to share with all of you how you can actually use magnetic separation to perform antibody and protein purification. So for some of you out there who are already performing antibody and protein purification, you may be more familiar with the traditional conventional method of doing so, which is using chromatographic methods. Okay, but you can also consider using magnetic-based methods for separation, because you can find that magnetic-based separation methods can actually help you speed up and simplify this entire process. Okay, so later on we will talk more about this, how it actually works, and the technology involved as well. But before that, let's start off with the basics. Okay, so firstly, why are pure proteins required? Okay, so pure proteins are actually very, very useful because you can use them for various applications. So for example, you can use pure proteins for performing analytical characterization. So let's say you are interested in studying a single protein. Okay, so you want to maybe determine the amino acid sequence of that protein, or you wish to perform some structural or functional studies on that protein. So if you are interested in studying just one protein, then definitely it helps if you have a very pure protein sample, okay, for analytical characterization. But of course, pure proteins can also be used in other applications. So for example, in the industry, okay, for example, in the pharmaceutical industry, you will see that a lot of big pharma companies are now trying to come up with antibody drugs. So they use antibody drugs as a therapeutic drug to help cure different types of diseases. So when they actually generate antibodies, it has to be purified, okay, to a certain level before these can be actually used as a therapeutic drug. Okay, so not just for pharmaceutical applications, but even antibodies or enzymes that you find in diagnostic kits. So these antibodies or enzymes that you actually use in these kits will have to be at a certain level of purity so that they are able to carry out their biological activity. Okay, and you don't get any interference from other things that may hinder their activity. Not just in the pharmaceutical industry, but also even in other industries like the food industry. So there may be some processes in the food industry that makes use of enzymes, okay? And these enzymes, again, will have to be of a certain level of purity before they can perform well, okay? So again, not just in food, but even in other industries like in the cosmetics industry and so on, pure proteins are often required, okay? So they are very important. Okay, so now that we've looked at why are pure proteins important, now let's look at how are proteins actually being purified. Okay, so usually when you have a sample, okay, your sample will contain your protein of interest, and it may also contain other contaminants. Okay, so other potential contaminants, for example, other biological molecules. It could be other lipids or other proteins that you do not wish to analyze. It could be that your sample contains things like debris, salts, reagents. So these are examples of other components that you wish to eliminate or to remove from your sample, okay, other than your protein of interest. So how do you actually purify your protein of interest from other potential contaminants? So what you need to do is to identify some physical characteristic of your protein that is different from the other potential contaminants. So you need to find a unique property of your protein and use that property to actually purify your proteins from the sample. Okay? So the method that you choose, the purification method, would then depend on the level of purity that you require as well. So if you look at this list of a few different types of purification methods, right? You can see, for example, you can perform things like centrifugation. So centrifugation will purify your proteins based on the differences in their densities. You can perform salting out, which will purify your proteins based on the differences in their solubilities. you can perform electrophoresis, which will again separate proteins based on the differences in their sizes and their charge. If you use a resin-based method for purification, depending on the chemistry of the resin itself, you can separate your proteins based on the differences in their size, their charge, their hydrophobicity, or based on affinity. Okay, so again, depends on the type of resin that you choose. And then finally, you can also use magnetic beads to perform affinity-based types of purification. Okay, so these are just some of the examples of the different types of purification methods that you could consider. But of course, they are all very different. Okay, generally, the affinity-based methods tend to give you much higher purification purities because affinity-based methods tend to be a lot more specific to your protein of interest. Okay, so if you really require an extremely high level of purity, likely you will need to perform affinity-based types of purification. Okay, and at GenScript, yes, we do have solutions if you are interested in resin-based method of affinity purification or magnetic-based methods of affinity-based purification. Okay, so now let's look at the differences between resin-based methods and magnetic beads-based method of affinity purification. So if we are talking about affinity-based methods, okay, generally there are two types. One is again resin-based. The other one is magnetic-based. Okay, so what is the main difference between the two? So the main difference comes in the type of bead that you actually use for each of them. Okay, so for resin-based method of purification, you would use usually agarose resins. Okay, so these agarose resins will be packed into columns, and then you use these columns for your purification. Whereas magnetic-based methods of separation would then make use of magnetic beads during the purification step. Okay, so that's the main difference. And if you look at the beads themselves, so agarose beads tend to be a little bit bigger in size. Okay, so they tend to be about 50 to 150 microns in size, whereas magnetic beads can be a little bit smaller, so about 1 to 100 microns. Okay, so this is good because if your beads are smaller, this means that overall, collectively, you have a larger surface area to volume ratio. Okay, so this can actually give you a higher binding capacity because they are smaller in size. But of course, if you were to look at the different beads now, okay, agarose beads tend to be very porous. So what this means is that this is, for example, an agarose bead. Okay, you can see this blue circle here. So an agarose bead will have a lot of pores from the surface of the bead all the way to the interior most of the bead. There will be pores. So all these little holes or pores extending from the surface all the way into the interior most. Okay, so this actually helps to increase the surface area to volume ratio as well. Okay, so with agarose beads, you tend to find that it can give you pretty high binding capacities because of this porous nature. Okay, however, of course, if your target protein is extremely large, then they may not have access to the inner binding sites. But I would say for general, most proteins, they should be able to actually diffuse into the innermost site of the agarose resin. For magnetic beads, then it's quite different. So magnetic beads are generally non-porous. Okay, so you see it looks something like this. So with that, it may give you slightly lower binding capacity because it's not so porous. But again, remember that magnetic beads can be also smaller in size. So again, that gives you collectively higher surface area to volume ratio as well. So in both cases, right, I would say they both give you very good binding capacities. That is pretty comparable. Okay, but I would say the main difference between a magnetic bead and an agarose resin is the fact that a magnetic bead can be attracted to a magnet. Okay, so if you look at this, if you have a magnetic bead here and then you have a magnet nearby, then what happens is that this magnetic bead will be attracted to the magnet. But in the case of agarose resins, then even if you have a magnet nearby, it will not be attracted to the magnet. Okay, so this is a very important difference because it is due to this property of magnetic beads that actually helps to simplify the entire purification workflow. Okay, so now let's look at what is the difference in workflow between between using resins and between using a magnetic bead. Okay, so this is the workflow if you are performing a resin-based method for purification. So like I said, remember, if you are using resins to purify your proteins or antibodies, likely you will need to pack these resins into a column. Okay, so this is a column. You can see this image here. So this is a column, and the blue circles here are your resins. Okay, so your resins are packed into this column, and then on the surface of your resin, you have your ligands, which are bonded on the surface, and your ligands will be the ones that will be used to bind to your target protein of interest. Okay, so before you start your purification, you need to make sure that you have a packed column first, like this, and then you have your sample. So in this example here, you can see that the sample is a cell lysate sample. Okay, so you have some cells which you have been culturing, and the cells have been producing your target protein of interest. Okay, so once you think that the cells, have expressed a sufficient quantity of your proteins and you wish to perform the purification, then what you can do is perform your cell lysis. Okay, so you break open all the cells and expose your target proteins. So the target protein in this example is the one in pink. Okay, so this is the protein that you wish to purify. So once you have performed your cell lysis, so then what happens is that there will be a lot of large particles, a lot of debris, which are still present in your sample. Okay, so before you put your samples into your column for purification, you must perform this centrifugation and filtration step. Okay, so this step is to help remove the large particulates like debris that can be found in your sample before you actually load your sample onto the column, because you do not want your sample to clog up this column. Okay, so the problem with using columns for purification is that these columns can be prone to clogging up. Okay, because again, what you want to do is that when you put your samples into the column, your samples are supposed to flow through this column. Okay, so if you have any large particulates that you also put inside the column, then it will block the flow, and then you cannot perform your purification. Okay, so again, as long as you are using a column, you need to perform this centrifugation and filtration step to prevent your column from clogging up. Okay, so this easily takes about at least 30 minutes to an hour to perform this step. And then once you have done this, then put your sample, load it onto your column. And then what happens is that your target protein of interest, which is the one in pink, will go and bind to your ligand, okay, which is on the resin. And then the other components will flow through the column. And then, of course, there will be some components that will still be binding non-specifically to your resins. So what you need to do is to wash off, so add a wash buffer to actually wash off any non-specifically binding proteins or compounds, and to try and wash off as much compounds as possible. And then finally, when you think it's clean enough, then you add your elution buffer, which will then be strong enough just to elute out your target protein of interest. Okay, so this is the entire workflow that you would probably expect if you are using a resin-based method for purification. So what about magnetic-based methods now? So same thing, we start off with the same cell lysate sample, okay? But now, once you've lysed your cells, you do not need to perform this centrifugation and filtration step, okay? So immediately after you have lysed your cells, you can just put your sample in a tube like this. So you can see in magnetic-based methods, we do not use columns, okay? We actually use just a regular tube, okay? Put your samples in there. So this is your cell lysate sample. Just immediately after you lyse your cells, put it in a tube. Then add your magnetic beads to your sample, okay? So your magnetic beads, as you can see here, is the gray circles here. So these magnetic beads, remember, they are very, very tiny, okay? So what this means is because they are so small, when you add it to your samples, they will actually float inside your sample. They will not, they will not settle to the bottom. They will actually be floating and resuspended in the sample. And then what they do is that you just leave it there to incubate for, let's say, 1 hour, 2 hours. And then what they will do is that they will then bind to the target protein of interest. Okay, so in this example here, the target protein of interest is the one in green. Okay, so you will see that after you let your beads go and bind to your sample, then all the beads will bind to its target protein, which is the one in green. So for every gray circle that you see here, there is a green circle, which is the target protein of interest that it has bound to. And then once you think it's enough time, then you can just expose a strong magnet nearby. And then this magnet will actually pull your magnetic beads to one side, and then the target protein, of course, follows along. Then you can remove the supernatant, which will contain compounds that did not bind to the bead. Okay? And then again, perform your washing and elution. Okay? So then this final step then gives you your target protein of interest. Okay? So as you can see, right, in this entire workflow, what you can save in terms of your time would be the time taken to actually perform that centrifugation and filtration step. Because again, in magnetic-based separations, you do not use a column. So therefore, you do not need to worry about your column clogging up. So you can actually get away with not performing this centrifugation and filtration step. And not just that, but because we do not use a column, therefore you do not need to pre-pack your column at all or perform any equilibration of your column. So these are, again, some time that you can save if you were to use magnetic-based methods of purification. So this slide, it actually just summarizes some of the advantages and disadvantages of the different platforms out there. Okay, so if you can see, right, the first three platforms, so the gravity column, chromatographic methods, liquid handling systems. So these three platforms typically make use of resin-packed columns to perform the purification. Whereas the last one here, this system, okay, it performs the purification based on magnetic rods. Okay, so the first three are based on resins. The last one is based on magnetic beads. So let's look at the first one first. Okay, so you can choose to perform purification just based on gravity columns. So gravity columns look something like this picture over here. Okay, so it's literally what you do is that you take a column, okay, which has your pre-packed resins at the bottom here. Okay, so this is your column, and then you do not with this method you don't use any instrumentation at all. Okay, so the good or the advantage of this method is that you can do this entire procedure manually, and because of that, it has a very low cost. Okay, because there is no equipment, expensive equipment required. But of course, the disadvantages of this method is that because you are using a column like this, okay, again, so before you load your samples onto the column, you need to perform that centrifugation and filtration step. So you need to perform that sample clarification step first, okay, to avoid clogging up your column. So that's one. Secondly, of course, you can do this manually, but as you can imagine, right, the throughput will be quite limited because you are basically going to do this manually by loading your samples into the column, and then you wait for gravity to pull your samples through the column, and then next you add your wash buffers, wash buffer 1, wash buffer 2, and then again you have to wait for gravity to pull your samples through, and same thing for elution buffer as well. Okay, so it's going to take quite a long time to actually purify your sample. So throughput can be very limited. Okay, and manual labor can be quite tedious also for this. Okay, so because of that, a lot of people, they try to automate this entire process. And so one option that they can choose to do is to use chromatographic systems, okay, which look something like this. So these chromatographic systems, they can actually take these columns and then put it on the system, and then the system can help to automate the entire purification. Okay, so one of the advantages of chromatographic systems is that it, of course, automates the entire process, but at the same time, it actually gives users the option of performing gradient elutions. Okay, and this can be useful, especially if you are in the method development step. Let's say, for example, you are not sure what sort of elution buffer to use for your purification. So having gradient elutions can actually help you perform that method development and gives you that sweet spot where it gives you the best elution buffer to use. Not just that, but these chromatographic systems can also take in a very wide sample volume range. Okay, so you can actually tweak some of the sample loops or the pumps in the system in order to accommodate larger sample volumes. And these systems can also be very flexible. So some of them are modular, so you can actually change, remove certain components or add in certain components depending on your application. Okay? But of course, some of the disadvantages of chromatographic systems is that the samples, again, because you are using columns on these systems, so the samples have to be clarified first. Okay, so again, you cannot run away from performing your centrifugation and filtration step. And not just that, but these systems are highly sensitive to things like air bubbles or high pressure. So this means that anyone using these types of chromatographic systems will have to be properly trained and they need to know how to use these instrumentations. If not, there will be issues when they run it on these instruments. But one major disadvantage is that these systems also do have limited throughput because you can actually mainly process one sample at a time. Okay, so usually users will process only one sample at one go. So if you have 5 samples or 10 samples that you wish to purify, then you have to do them one at a time. So the throughput can be quite limited and the cost can be quite high as well. Next, we have liquid handling systems like this. Okay, again, it provides automation, which is great. Throughput can be quite high as well. Okay, and it can be quite flexible too. Okay, because it can be modular as well. But some of the disadvantages of liquid handling systems is that, again, if you are using columns to perform your purification, you need to perform that clarification step first, that centrifugation and filtration. And usually liquid handling systems cannot really process very large sample volumes. Okay, so usually sample volumes will be lower and the cost can be quite high as well. Lastly, we have a magnetic rod. So this is an example of a magnetic-based method for purification. Okay, so it's no longer using columns. So this system that you see here, it makes use of magnetic rods to actually perform the purification. And it helps to automate the entire process as well. And the throughput is pretty high. So you can see here, what you basically do is you put your samples in either 24 or 96 well plates. And then you also put your wash buffers and your elution buffers in here as well. Okay. So, the disadvantage of this method is that, again, it can only take in small sample volumes only because, as you can see, your sample is only restricted to a 24 or 96 well plate. And reagent preparation can be quite tedious because you need to aliquot all your wash buffers into each well of the plate. The cost could be also pretty high. And of course, you need to use magnetic beads with this device. Okay, so for those of you who may not be familiar, you may think that magnetic beads may seem pricey, but later on I will talk a little bit more about this as well, because at GenScript we actually do also have magnetic beads, but our magnetic beads can be reused multiple times. So with that, you can actually decrease your cost of purification in the long run. Okay, so later on I'll talk more about that. But basically, as you can see, right, these are different platforms that are out there. And I would say each of them has their pros and cons. Okay, but I would say the main disadvantage, as you can see, right, is that if you are using resins or columns for purification, then you need to perform that sample prep step. Okay, but as you can see, some of them also have very low throughput. For example, the gravity column, the chromatographic methods, they both have very limited throughput. Okay. And in the case of liquid handling systems and this magnetic rod, purification system, you can see that they can only handle small sample volumes. So these are, I would say, the main bottlenecks or the limitations of the current platforms that are out there now. Okay? So with that, I would like to then introduce you to this instrument that we actually have at GenScript. Okay, so this instrument is actually called the AMAG SA Plus system. Okay, so it looks like this. And this system can actually help to improve on the limitations that we just spoke about in the previous slide. Okay, so the AMAG SA Plus system, later on we will take a look at some of the specifications. You can see that it can actually help to increase your throughput and also manage to actually purify higher levels or larger samples of volumes. Okay? So later on I'll talk more about this, but basically this instrument is actually designed by both GenScript as well as Amgen. Okay, so they both came together to come up with this device to help users like yourselves actually perform antibody and protein purification using magnetic beads. Okay, so this is a system that can help you automate the purification process. But of course, if you wish to actually perform magnetic separation, you can also perform it manually. You don't have to use an instrument. Okay, so if you wish to perform it manually, you can actually use these magnetic holders like this. Okay, so these are holders which have magnets on there, and then you can actually just add your magnetic beads to your sample, and then process the samples manually. Okay, so this is how you can actually do it manually as well. So it's optional. If you think that you wish to perform this manually, you can. But if you think that you wish to automate the entire purification process, you can consider an instrument like the AMAG SA Plus system. Okay, so if with the AMAG SA Plus system, these are some of the advantages that you do not need to perform again centrifugation or filtration, because this is a magnetic-based method for separation, so it doesn't use a column. There is no column packing, no column equilibration required. It's able to handle medium-sized sample volumes, okay, so not just small sample volumes, but also medium-sized. And it's easy to automate for higher throughput as well. Okay, so if you wish to process more than one sample at one go, you can do so with this instrument. Okay, so before we look at, so this slide actually, it shows you the features of this instrument, but before I actually talk about the features, so this is how the AMAG SA Plus instrument looks like again. Okay, so it's a very simple instrument, as you can see. At the top of the instrument, it's basically the interface. Okay, so this is when you turn on the instrument, you will see that this interface will light up, and then this is basically where you tell the instrument what you want it to do. Okay, so your purification protocol, your regeneration protocol, everything will be done over here. Okay, so this is a touch screen, so you can basically just use your finger or use the stylus that is provided over here. You can use that to actually key in the variables that you wish to key in. Okay, and then the bottom part would be where you would place your samples. Okay, so this is a safety door. Okay, so all you just have to do is to open this door, and then you will see that there are 2 racks inside here. Okay, so each of these racks can actually hold 6 samples. So in total, you can actually process 12 samples at one go. Okay, so this is why it has higher throughput, because you can actually process up to 12 samples at one go. Okay, so this is basically how it works. Okay, so now let's look at the features. So some of the benefits of this system is that, of course, it helps you automate the purification process. So it helps to reduce manual labor required. It also eliminates the need for sample centrifugation and filtration, just because it is a magnetic-based method of purification, so no columns used. It gives you a closed environment to minimize endotoxins contamination. So for those of you especially who are concerned with endotoxins levels, some of you may be perhaps purifying antibodies, so endotoxins may be of a concern to you. So having a closed environment, because as you can see, your samples will be in the instrument the entire time, then this helps to minimize endotoxins contamination from the environment. Okay. And one important characteristic of this instrument is that you can use our reusable magnetic beads with this instrument. And this can then help you lower your overall cost of purification as well in the long run. Okay, and these are just some specifications of the system. Like I mentioned, it can purify up to 12 samples at a go. Okay, and each batch of 12 samples can take about 45 minutes to complete. Okay, and you can use up to 5 buffers for purification at one time. Your sample volumes range from 1 mil to 50 mils, okay? Because as you can see in this instrument, it is compatible with 50 mil tubes only. So remember I mentioned that there are 2 racks in the instrument. So these 2 racks, they can hold 50 mil tubes only, okay? But so you may have a concern. You may think that what if I have 100 mils or 500 mils of samples? Can I process it with the MEG SA Plus instrument? Yes, you can. It's just that you need to do one sample preparation step before you can load your samples onto the instrument. And later on, I will talk more about that. Okay, so don't worry. Even if you have samples like larger volumes of samples, like 500 mils, 1 liter worth of samples, you can still process it using the MEG SA Plus system. The minimum elution volume is 0.4 mils. Okay. And it can purify up to 80 mix of antibodies or proteins per sample, depending on the bead that you are using. And the software here lets you save up to 50 different purification protocols. Okay, so these are just some of the specifications of the instrument. And as you can see, it is a magnetic-based method of purification. So just be aware that this also comes with a strong magnetic field because there are strong magnets associated with the instrument as well. So this is the entire instrument setup. It looks like this. Okay, so the instrument will also come with some accessories. For example, it comes with all these bottles, okay, as well as the tubings and the rack that you see over here. So at the side of this, the right-hand side of the instrument, you will see 8 different ports. Okay, so each port will be linked to a bottle. Okay, so the first 5 ports will be linked to your 5 different buffer bottles that can be used for your purification. Okay, so these 5 buffers will be the buffers that you use for washing of your beads and also performing the final elutions as well of your protein. And then the 6th and the 7th port will be linked to buffers that will be used to wash the nozzles which are on the instrument. Okay, so there are nozzles inside the instrument that you cannot see over here. Okay, so these nozzles will be used to go and pick up your samples and to also dispense your buffers into your samples. So before, when it goes from one sample to another, it will wash itself in between samples because you don't want any cross-contamination to take place. So this is why we also need to connect a couple of wash buffers for washing the nozzles here. Okay, so that will be ports number 6 and number 7. And then the very last port number 8, will be linked to a waste bottle, okay, a 5-liter waste bottle, which will collect all the waste buffers. Okay, so this is the entire instrument setup. I won't go into the details about the software itself, but the software, it's really pretty easy to use. Okay, so like I mentioned, it's actually a touchscreen software. Okay, so once you turn on the instrument, then you will see the software will pop up. Okay, so this is how the software actually looks like. Again, I won't go into the details, but as you can see, you have 5 tabs that you have at the top. Okay, the first tab is called the pre-treatment tab. And so just to summarize, this pre-treatment tab, what it does is it gives you the option of cleaning your system before you start the actual purification. So pre-treatment tab is for cleaning. So you can clean your system with some sodium hydroxide to minimize the endotoxin levels if that is of concern to you before you actually perform your purification. You can also turn on the UV lamp to sterilize the system if you wish to further sterilize it before you start your purification and so on. So this pre-treatment tab is to clean your system. Then once you think it's clean enough, then you can click on the Processing tab, and then you come to this page. And the Processing tab is basically for you to tell the instrument exactly what is your purification protocol. Okay, so you can select the positions of your samples that you put in the instrument, and then tell the system what is your purification protocol over here. Okay, so once you've input all the values inside, then you just click play, and then the instrument will perform the purification. Okay, so then once the purification is done, all you need to do is then go to the instrument and collect your final purified samples, and then what is left in each sample tube will be your magnetic beads. Okay, so then remember, I mentioned that we have magnetic beads that can be reused many times. So what you want to do next is to go to the stripping tab. Okay, so this stripping tab is for you to clean your magnetic beads. Okay, because after purification, your magnetic beads may contain some other compounds like proteins that can still be non-specifically binding onto the magnetic beads. So you want to clean it, to strip it, to make sure that it is very clean, and then you store it, and then you can reuse these beads for subsequent purification experiments as well. Okay, so this stripping tab is for you to clean the magnetic beads, okay, before you store them and reuse them. Okay, so these are the three main tabs that we have. And then the settings and system tab mainly is actually used by our engineers when it comes to performing some servicing. Okay, so as you can see, it's a very simple software to actually use. Okay, so and in terms of results, of course, this is very important. This is actually some results that was obtained from a customer where they actually compared the recoveries that they were obtaining when they used our MAG-SA system versus a typical chromatographic system. And they actually purified different antibodies on these two systems. And you can see that the recoveries are actually extremely similar. Okay, whether you use our MAG-SA device or typical conventional chromatographic systems. Same thing, we also have some data showing you antibody recoveries between the MAG-SA system, which is the one in yellow, and an automated liquid handling system now. Okay, as you can see, again, I would say it may seem like the MAG system has a higher recoveries, but I would say conservatively that the MAG-SA device has very comparable recoveries as compared to an automated liquid handling system as well. Okay, so recovery is important, but of course, purities are also important. So as you can see, these are some of the results when you run your purified samples on a gel. You can see that whether you purify your samples using a conventional resin or an MAG-SA device, you can see that the purities that are obtained are very similar as well. But of course, with the MAG-SA, then it comes with some benefits that, again, you don't have to perform that sample prep step, that centrifugation and filtration. And you also do not need to pre-pack your columns, and you can actually increase the throughput of your purification. Okay. So again, these are just some of the things that we compared. So between two different MAG-SA systems, we see that the purities that we obtain between them are very consistent, okay, about 97 or 98%, which is similar to that of a resin-based method of purification. And in terms of the endotoxins levels, it's also all very low. Okay, so that's actually just giving you an introduction to our MAG-SA Plus system. So that's what you would probably use if you wish to automate this purification process. But of course, with the MAG-SA Plus instrument, you will then need to use magnetic beads with the system. Okay, so at GenScript, we also do have a lot of different types of magnetic beads available. So if, let's say, you are interested in, for example, antibody purification, so these are some of the magnetic beads that we have. Okay, we have protein A magnetic beads, we have protein G magnetic beads, we have protein AG, we have protein L, we have this MAG protein A mag beads as well. So I won't go into the details of all the different beads, but what I want to highlight here is actually this MAG protein A magnetic beads. Okay, so this MAG protein A magnetic beads, as you can see, its binding capacity is extremely high. Okay, it's more than 40 mix per mil. And I believe this is actually the highest binding capacity in the market right now. Okay, so it can bind to a lot of your IgGs. And one very important aspect of this AMAG protein A magnetic beads is that they are alkaline tolerant. Okay, so this means that you can actually use an alkali, for example, sodium hydroxide, to actually clean these beads, and therefore you can reuse these beads multiple times. Okay, so these AMAG protein A mag beads can be reused up to about 40 times. So this is what we have tested in our labs as well. Okay, so as long as you see the word AMAG in front of our magnetic beads, it means that these beads can be reused multiple times. Okay, so this is something that is really, really pretty useful, I would say. Okay, so if you're considering performing antibody purification, this is a mag bead that you can actually consider, because again, can reduce your cost of purification in the long run. So we also did some studies on these magnetic beads as well. So this is the AMAG protein A magnetic beads. So we actually compared these beads with beads from another vendor. Okay, so you can see that the AMAG protein A mag beads are the ones on the right. The beads on the left are from supplier B. Okay, and we put these two beads on top of a magnet. Okay, so at 0 seconds, you can see that all the beads are still suspended. Okay, but then after you leave it there for 6 seconds, you can see that our beads start to get attracted to the magnet, which is what you expect. Okay, but the beads from supplier B take a longer time to actually respond to a magnet. So that means that if let's say, for example, if you were to use beads from supplier B to perform your purification, that it may take a really, really long time, okay, for it to go to completion. Okay, so this is one result that we have. And then we also tested out our binding capacities, okay, of our beads. You can see that as you increase the antibody concentration in the sample, then the binding capacity also increases. Okay, so this is because we are able to have very high binding capacities. So therefore, as you increase the concentration of your IgG in your sample, the recoveries of your it starts to increase proportionately as well. Okay, whereas if your beads have very low binding capacities, then it tends to saturate out like this competitor brand. Okay. We also tested our beads to make sure that they are alkaline resistant, so that you can use alkali to clean the beads, okay, over and over. And you can see that even if you clean the beads for 10 times, 20 times, 30 times, you can see that the binding capacities is still maintained. It's only at about 40 cycles of cleaning of these beads that we start to find that there is a drop in binding capacities. Okay, so therefore, we only suggest that you reuse these beads up to about 40 times, because anything more than 40 times, you may observe a drop in binding capacities. Then that's when your recoveries may be affected. Okay, and lastly, we also checked for protein A leaking, because protein A is the ligand on your magnetic beads. So we do not want protein A to be leaking or bleeding off the beads. So we checked for protein A leaking off the beads, but as you can see, there is very minimal in the nanogram scale. Okay, so very little protein A is leaking off the beads, which is great. Okay, and if you wonder, how clean are your beads after regenerating them? So as you can see here, we also did some tests. So lanes 1, 3, 5, 7, 9, 11, so the odd number lanes, contain your magnetic beads, which contains residues before regeneration. So this is after you perform your purification, but before you perform regeneration of your beads. Okay, so we ran these beads on this gel, and you can see that the beads still contain a lot of proteins bound onto its surface. Okay, so these are non-specifically binding proteins after we perform purification. Then the lanes 2, 4, 6, 8, 10, 12. So these lanes are the magnetic beads after performing that cleaning, okay, after performing that regeneration. You can see that you do not see any proteins on these beads anymore. Okay, so this means that using the default regeneration protocol that we have on the MagSA system can help you remove residual proteins on the beads. Okay, so then you can store the beads and then reuse them for the subsequent purification. Okay. Okay, so that those are for the beads that we have for antibody purification. We also have beads for tagged protein purification. So if you have any proteins that are tagged, we also do have magnetic beads for you to actually perform the purification. So if you have his tag proteins, you can use our nickel charge magnetic beads or our MAG nickel magnetic beads for purification. And if you have, so these are other beads that we have also for other tagged protein purifications. Okay. But the main thing that I wish to actually talk about on this slide is actually our MAG nickel magnetic beads. Okay, so our MAG nickel magnetic beads, again, you can see the word MAG in front. So this means that these beads are highly reusable. Okay, so if you have any his tag proteins that you wish to purify from your samples, these beads, again, they are alkaline resistant. Okay, so you can use sodium hydroxide to wash these beads and regenerate them. And you can even use them for up to 100 times. Okay, so this is something that is really very beneficial. And these beads are also slightly resistant to EDTA as well. So we did some tests also. As you can see, our MAG nickel magnetic beads, we actually tried to wash them many times with sodium hydroxide to regenerate these beads. You can see even after 50 cycles of purification and 50 cycles of regeneration, 100 cycles of regeneration, 150 cycles, you can see that the capacity of the magnetic beads is still being maintained. So even at 100 times of reuse, 150 or even 200 times, it seems that you can actually use these beads many times. So what we recommend is that you can actually reuse these beads over 100 times. Okay, so it's again highly reusable and therefore would overall reduce the cost of your purification. Okay, so those are the beads that we have. And like I mentioned, if you actually have larger volumes of samples that you wish to purify, for example, if you have 500 mils of samples that you wish to purify, don't worry, you can still purify that on the MagSA Plus system. So all you need to do is to use an accessory, for example, this MAG magnetic wand, okay, which looks like this. So all you need to do is use this wand, magnetic wand, and collect all the beads which are incubating in your sample. So this sample, for example, it's maybe 500 mils of sample. Collect all the beads using the wand, like this, okay, and then transfer these beads into a 50 ml tube, and then resuspend them or reconstitute them in maybe 50 mils of a buffer, and then put it onto your MagSA Plus system for processing. So that's all you need to do. Okay, so don't worry, even if you have larger sample volumes, you can do this step first, collect all your beads, put it into a 50 ml tube, and then process it on the MagSA Plus instrument. Of course, you can also use like a MAG magnetic block like this, okay, to actually help to collect your beads onto the bottom of your bottle here. And then you pour off the supernatant and then reconstitute the beads in a volume of less than 50 mils. And then you can actually load it onto the MagSA Plus system to process as well. Okay, so this is the entire complete solution that we actually have. For any of you who are interested in performing magnetic-based method of purification to purify antibodies or proteins, you could do this either manually, or if you wish to automate the process, we do have a system as well. So for manual solutions, we actually do have things like magnetic mini racks like this. So if your sample size or your sample volume is very small, then you can actually use a mini rack like this to perform your manual purification. Okay, so this rack can actually hold your 2 ml Eppendorf tubes over here. Or if you have a larger sample volume of up to 50 mils, you can use a larger magnetic rack. So these ones will hold 15 or 50 mil tubes. We have a magnetic box which can hold 50 mil tubes as well. So all these are solutions that you can use if you wish to perform this manually. Okay? But of course, if you wish to actually automate the entire process, then we have the MagSA Plus. Okay, and again, if you have samples larger than 50 mils, you can use our magnetic block or our magnetic rod to decrease your solution size first before you load it onto the MagSA Plus system for purification. Okay. And we also do have some plans to actually come up with another instrument that can actually take in larger volumes of samples for purification. So this is called the Antibody sample. So it can take up to 2 liters worth of your sample volumes. But this is still in progress and we have not released this instrument yet. Okay, so before I end, I just also want to mention that apart from protein purification, we also do have other solutions for protein analysis. So what we talked about is this particular system that we have, which is the MagSA Plus system, which is used for protein purification. Okay, but for example, subsequently, after you have performed protein purification, sometimes you may want to then do a gel run to actually check for your purities. We also do have some consumables as well as instruments for you to perform that. Okay, so we do provide precast gels like this as well. And we do have some devices that you can use to stain your gels. So for example, this E-stain system, okay, it's actually a protein staining system where you can basically, once you run your gel or your proteins on your gel, you can load the gel onto this instrument and it will help to stain for all your proteins using Chromatographic blue. And this is done within 10 minutes. Okay, so it's a lot faster compared to the conventional manual method of performing this staining. We also have the E-Blot system here, okay, which will help to transfer your proteins from a gel to a membrane. So if let's say you are performing Western blot, okay, so this is actually a transfer system that is really great because as you can see, its transfer efficiency is equivalent to the gold standard wet transfer method, but it transfers all your proteins within 9 to 17 minutes. So it's a very fast wet transfer system. Okay, and lastly, we also have this Easy West Light automated Western system, which then helps you complete your final step in the Western blot protocol, where it helps you automate that blocking step, that incubation of your primary and secondary antibodies, as well as all wash steps as well. So you can automate all those steps using this single instrument over here. Okay, so this is what I want to introduce to you today. And just my very last slide, so it's a summary slide. Basically, what we've gone through is that the MagSA Plus instrument, it can help you automate the purification process. So it shortens your purification time as well, and it saves you some labor. It helps you purify 12 samples at one go, okay, in 45 minutes. Typically, that would be done in 45 minutes. No proprietary buffers or consumables are required to be run on this instrument. We also do have a variety of magnetic beads which are available that you can use on this MagSA Plus system, or you can choose to use them manually as well. So for example, we have the AMAG Protein A magnetic beads, which are alkaline tolerant. So you can wash them with sodium hydroxide and you can reuse them up to 40 times. And they have a very high binding capacity. I believe it's one of the highest now in the market. If you have his-tag proteins and you wish to actually purify them, you can consider also the AMAG nickel magnetic beads. They are also alkaline tolerant and they can be washed with up to 1 molar of NaOH and used up to 100 or even 200 times. Okay, and they are also slightly resistant to chelators like EDTA and DTT. If you have very large sample volumes, for example, 500 mils or 1 liter, you can reduce them to 50 mils or less first using the accessories that we have, like our magnetic wands or magnetic blocks, and after which you can put these samples on the MagSA Plus to purify them. And we basically also have other solutions for not just protein purification, but also for Western blot or gel staining and so on, like I mentioned in my last slide. Okay, so lastly, we do have also free samples of magnetic beads available. So for any of you who actually wish to explore using magnetic beads for performing your antibody or protein purification, just do take note, you can reach out to us and we can actually give you some free magnetic beads for you to try out as well. Okay. So yeah, with that, I've come to the end of my presentation. And thank you so much for your time.